Hello, General Biology, and welcome to screencast number 2 in our Chapter 1, Section 1.1. And we were talking about some of the scientific methodology. So where we last left off, we were looking at these two different grasses, and we are going to infer in order to be able to form a hypothesis. So the scientists, the researchers looking at this, realize that there is something that is limiting this grass growth in the marsh. And based on what they know about salt marshes, they can hypothesize that whatever is missing is what is impacting the growth. So when we learn in chapter 3 or 4, we're going to be learning about the nutrient cycles, and one of those nutrient cycles is the nitrogen cycle. And nitrogen is something that is found in fertilizers. So based on that knowledge, they're going to say, well, maybe one of these areas is limited by the amount of nitrogen that is available within the soil and the water of the salt marsh. So based on what they know about what plants need to grow, knowing that nitrogen is a fertilizer, that it can help plants grow, maybe that's what the problem is in this area. So the next step, once they make that hypothesis, they're going to be able to design a controlled experiment. I want to flip back here because I want you to do something for me this year. And this is something, if you have me in other classes, we won't do this format, but we are going to do it in this particular class. When you do a hypothesis, I want you to write hypotheses in an if-then statement. An if-then statement is cause and effect statements. So what we think the cause is and what we think the effect is going to be as a result of that cause. So I can say, for this example, if the marsh grass in location A is limited in nitrogen, then it's not going to be able to grow efficiently. Can I test that? I can, because I can check if there's nitrogen available in location A. So it's cause and effect. So every time you form a hypothesis, I want you to put it in an if-then statement. If the grass is green, then elves painted it green. I can test that. I can test that cause and effect. Okay? So based on that, we can now design a controlled experiment. And a controlled experiment is going to test that scientific hypothesis. And one of the things that we're going to have to consider is what our variables are going to be. We've got multiple variables. Variables are things that can change, or factors that can change. So temperature can change, light can change, time can change, availability of nutrients can change. There's a lot of things that can change. So we're going to control some of those variables so that limits us to only one variable that we're testing at a time. And that's key. You only want to test one variable at a time. One test, one variable. We don't want to have multiple variables because then we're not going to know which variable is causing the change. So, I'm not going to worry about sunlight or the amount of water or the presence of carbon or the pH because that's too many variables. I'm just going to look at nitrogen. And if I find that nitrogen's not having an effect, that there's the same amount of nitrogen as in A as there is in B, then I can go to pH, then I can go to temperature, then I can go to the chemistry of the water. So I only want to focus on that one variable at a time. So a hypothesis needs to be tested by an experiment. We only want to test that one variable. All of the other variables then, what do we do with them? We keep them unchanged, or we keep them controlled. So here's where some problems come in and people get this confused. This is going to be a controlled experiment. It only focuses on that one variable at a time, and it keeps all of the other variables the same. Make sure that you write that somewhere down in your notes, because when we talk about what a control is, what winds up happening is a lot of people start to confuse a control with a controlled experiment. A controlled experiment, one variable. We're just testing one variable at a time. A control is going to be something we use for comparison in that experiment. So they're two different things, and I guarantee you that in the beginning they're going to confuse you because of that word control. When it's controlled experiment, it's that thing you're changing, that one variable, and the control is what we're going to use for comparison. And we'll talk about that a little bit more. So let's take a look at why it's important to control those variables. And as I said, if there were several variables that were being changed, the researchers wouldn't be able to tell which variable was going to be responsible for the results they observed. So they focus on one variable that they're going to change. And that one variable that they're going to change, we're going to call that the independent variable. And you might hear that referred to as manipulated variable. For me, I tend to say manipulated more than I say independent, because manipulated, it tells me when you manipulate something, you're changing it. So the manipulated variable, if I can say that, is what is going to be deliberately changed. What am I changing? Okay, so let's take a look at, we'll go back with my grass example. So if I want to find out if the grass is green, what can I change? I could take a look at the grass underneath the microscope, or I could put it in a container to make sure that no elves could get to it. So that's what I'm changing. I'm only changing one thing. So in our marsh grass, what are we going to change? We're going to change the amount of nitrogen. We're going to see what kind of effect nitrogen has on the grass. We might give one grass more nitrogen and see what happens, another grass less nitrogen, and then keep the normal amount of nitrogen that they should have, so that we can see. But we're just changing that amount of nitrogen. We're not worrying about anything else. We're not worrying about the light. We're not worrying about the temperature or the amount of water or anything like that. It's just that nitrogen. So what happens as a result of the change? Well, our response is, as a result of that change, is going to be called the dependent variable. Or I like to say the responding variable. It's the response to the change. So if I notice that the grass that was given more nitrogen grows taller than the grass that was given less nitrogen, I can see that nitrogen plays an important part in the growth of that grass. So it's the response to that change, or the dependent variable. So typically here, this is where I said you kind of get this confused. An experiment is divided into a control and experimental groups. So the experimental groups are what you're going to be changing, the amount of nitrogen. In our example, a high amount of nitrogen and a low amount of nitrogen. And we're going to have a control. And that control is going to be, and I don't know why my pen just wrote that way, under normal conditions, we're not going to change anything. It's going to be the normal amount that we should have. And we're going to call that our control group. It's exposed to the same conditions as the experimental group, except for the independent variable. So let's go back to the idea of a controlled experiment. We're only changing the amount of nitrogen. We are keeping the temperature the same, the amount of sunlight the same, the amount of water the same. So those conditions are staying the same. We're just adding nitrogen. So what we're going to do is we're going to find out what's the normal amount of nitrogen. And the normal amount is going to be our control group so that we have something to compare it to. Let's try something a little bit maybe easier. So let's say that I have 3 different plants and I want to see how light affects the plant of the growth. So I'm going to put one plant in a dark closet, another plant in the bright light, and another plant in the regular room light. The one in regular normal room light is going to be my control. And my manipulated variable, my independent variable, is going to be the change of intensity of that light, the dark versus the bright light. And then whether the plant grows or not is going to be my responding variable. So that control is in normal conditions. So a scientist is going to set up several sets of a control and in experimental groups to try to reproduce or replicate their observations so that they can see the effect that the variables have. So let's go back to our graphs that we're seeing here. So here they have two groups. They have the control group that you see right here and the experimental group that they have right there. In the control group, they're not going to add any nitrogen. They're going to keep it at the normal amount. And in the experimental group, they're going to add nitrogen to it because that's the fertilizer. So this is the change. This is what we're changing in my messy writing here. That's what we're changing. So that's going to be the independent variable or the manipulated variable. And then we're going to look at the growth. And if this grows higher, and we can tell it grows higher because we can compare it to the control group, then we'll be able to tell if the nitrogen is a factor. So here we go. We perform our experiment, and now the next thing that we need to do is collect the data, and we need to analyze that data. So we're going to collect the data by measuring the height of the grass after a period of time. And that measurement is going to be considered quantitative data because we can tell how much is there. We're also going to do qualitative data. Remember I said that qualitative data describes it, and we're going to look and see, how dark green is that color of the grass. Oh, look, see here, there's something that's in there that's a real dark green. Do we have any brown? And we're going to write that qualitative data down, as well as we're going to take our quantitative data, and that's our data that we can measure. We're going to measure it. We might even count how much grass we have there within a specific area. And we're going to collect that data. And next we're going to analyze it. And that analyzing means we're going to compare these two things. Was there a change? What was our response? Was there a change? Did it increase in size? And that's what we're going to look for. If it did increase in size, can we say adding nitrogen increased the productivity of the grass and it grew? We can, and that would be what the analyzing the data would mean. So quantitative data, as I said, is going to be numbers. You're going to get those in measuring. Examples would be the number of plants, plant sizes, the growth rates. All of that can be put in numerical form, taking the temperature of the soil, anything like that. Qualitative data is more descriptive, and it involves the characteristics that cannot be